everyone, welcome to our channel Biology for All. In this video, I'm going to talk about topic 6.1, that is digestion and absorption, and D-2 digestion. And in this, we are going to talk about the skills related to these two topics. So the first skill that we're going to talk about is the identification of tissue layers, that is the transverse section of the small intestine, as we look under a microscope. Now this is the light microscope we are talking about, and so we can see a micrograph, or we have to identify it from a micrograph. Now before we do that, let us first see how the transverse section of the small intestine looks like. So here, this is the transverse section of the small intestine, which has an outermost layer made up of connective tissue that is called as serosa. Next to that is a layer of muscles, that is the smooth muscles. Now this smooth muscles has two layers. The outer layer is longitudinal muscle and the inner layer is made up of circular muscle. So together it forms the muscularis. Inside that you have the submucosa. Now the submucosa is a layer where there are blood vessels, nerves, and the glands of these intestine which is present. The innermost layer that is next to the lumen, so lumen is in the center here, and the innermost layer to that is a mucosa, and these mucosa shows folds which then forms the villi. So here let us now see how it looks like under a microscope. So here this is how it looks like. We can see the villi here, and these are the different layers that are present, the mucosa and the muscularis and the serosa layers that are present. Now let's have a look at a little more detailed in that. So here you can see now this outside layer, this is what we talked about, the connective tissue layer, that is the serosa. Then you have the muscularis layer. So in this, the outer part here is the longitudinal muscle and the inner part here is the circular muscle. Then the submucosa and the mucosa. Like I said, the mucosa has those folds that form the villi. Another picture, if we can see, this is also a section of the intestine. Now here you can see the muscularis layer, the submucosa, and the mucosa which is forming the villi. Another section that we can see here, the cells. So here in this picture, you're able to identify the longitudinal muscle and the circular muscle also. So this entire layer here is a muscularis. This is the serosa, which is the outermost. So the layer of muscles which are outside is the longitudinal muscle, and the one which is inside is the circular muscles. Then you have the submucosa and the villi in the mucosal layer. So you may be asked to identify these layers. So you should be able to identify that the outer one is the serosa, then you have a longitudinal muscle layer, then the circular muscle layer, then submucosa and the mucosa, which forms the villi. So this was how you have to identify the transverse section of the small intestine. The next one is D2 skill 1, where you have to identify the exocrine gland cells that secrete digestive juices, and in that you have to try and identify the features that tell you that this is an exocrine gland. So first let us look at the exocrine gland here. Now this is pancreas is an exocrine gland. Now pancreas has both endocrine as well as exocrine glands. So the endocrine part is the islet of Langerhans, which secretes the hormones, insulin and glucagon. Now, apart from that, there are these acinar cells which are present. So this is the acinus, which has a duct that leads to the main duct, that is a pancreatic duct, which opens into the duodenum. And next, we also have to see the villus epithelium that absorb digested food from the electron micrograph. So that will be in the small intestine. So right now we're going to talk about the pancreas. So here the acinar cells and their secretions, which then go through the duct into the pancreatic duct. So how does this look like under the micrograph? So this is how the acinas look like. This is the acini. So you can see here these circular structures, these are the acini. So these structures what you see here, that is what is the acini, which have cells. And those cells will produce the secretions, that is the pancreatic juice. Now let us see the actual structure here in the form of an electron micrograph. So here we are now seeing the electron micrograph, unlike the previous one where we saw under a light microscope, that was just a micrograph we had seen. Now in this we are able to see different structures inside the cell, since it's an electron micrograph. So being an exocrine gland cell, obviously it is going to be secretory in function. So because of the secretory in function, there will be specific organelles for that. So one of the organelle that is responsible is the endoplasmic reticulum. We know that endoplasmic reticulum is for protein synthesis. So these are the endoplasmic reticulum. You can see it in this micrograph also, the electron micrograph also. Then there are mitochondria present. So you can see here, this is the mitochondria that are present. These are also mitochondria. Here you can see the mitochondria here. Same way, in this also you can see the mitochondria in these parts. Then the nucleus, this is the nucleus, and other than that there will be the Golgi complex. So this here you can identify this is the Golgi complex, and these vesicles, these are the secretory vesicles. So these secretory vesicles then are released into the lumen here. So you can see this part is the lumen. So this is one cell. You can see the mitochondria here. So this is one cell entire, and in that way there will be many cells which open into the lumen. That is, their apical surface is towards the lumen, and there they will release the secretions. Now in this case the lumen is here. This is the lumen. So these secretory vesicles will release the secretion into the lumen here. So this is the electron micrograph of the pancreatic cell, acinar cell, which is releasing the pancreatic juices, which then move into the intestine, that is in the duodenum, and there it helps in digesting. Now the intestine is further adapted for absorption. So for that, what are the things that are there? The main thing is the microvilli that we can see here, the microvilli that increases the surface area. Other than that, there are these structures here which are the tight junctions. So these are the tight junctions which keep the cell or hold the cell together. And then these microvilli, like I said, which increases the surface area. Apart from that, even these cells show a large number of mitochondria for active transport. To absorb glucose and amino acids, they require active transport. So there is a lot of mitochondria present here. And apart from that, there are these vesicles which are present. These are the pinocytic vesicles. So these pinocytic vesicles are the ones which help in taking up substances in liquid form. So that is the pinocytic vesicle. So this is how you identify the epithelial cells and how they help in absorbing the digested food with the help of these structures. So this is about the skills related to topic 6.1 and topic D2.